Hi, welcome everybody. I'm Waqas. I'm a third year internal medicine resident at NYU in the clinical investigator track and starting oncology fellowship at U Chicago this upcoming summer. I'm excited to bring you our next episode of the Oncology Data Advisor podcast. This week we'll be having a feature from Leif Honda, who's the Chief Innovation Officer at TriMetis, which is a company dedicated to automating the workflow and process through AI for different pathology services. Really excited to talk to you today, Leif. If you don't mind, just give us a short introduction about yourself and maybe just tell us a little bit about what TriMetis does and just the reasons for why it got started and how it's improving biobanking.
Yeah, thank you, Waqas. Again, my name is Leif Honda, and I'm the Chief Innovation Officer at TriMetis. We essentially have been in the business of handling specimens for research for over 20 years. And through that process, we have improved how we do things by the use of leveraging technology. And those technologies include artificial intelligence, workflow automation, and robotics. And so we are investing heavily in this space in order to really ultimately make life easier for clinicians and researchers. Behind all that is to try to accelerate the validation and approval of tools that clinicians can actually use on a daily basis. And I can go into that a little bit later.
Awesome. Thanks for sharing that. I also read that you have an economics background. I majored in economics myself, so glad to see someone with a similar sort of training in mind. So I want to sort of talk a little bit about the TriMetis Computer-Assisted Pathology platform that's been developed. I attended the Oncology Precision Medicine AR seminar last month, and one that I talked about in terms of bioethics, a lot of the training data for AI companies comes from three different states, Massachusetts, California, and New York. A lot of the biotech innovations happening. So tell us a little bit more about, how tCAP is trained, who trained it, and sort of what it encompasses.
Yeah, so tCAP came from a place where essentially we in research needed more detail on what was in the tumor tissues that we were using for research. So it wasn't just a cursory overview of what the pathology and the diagnosis was, but it was actually the morphology and the information contained in the tissue. And that was telling us more about whether those tissues were usable or not for certain studies. And we would screen and fail lots of tissues trying to find explicitly these perfect cohorts of patient data and tissues in order to do discovery of novel biomarkers, therapeutic targets, and really landmarks and things that could tell us something about the disease state and the change in disease state. That was a very difficult thing to do. And at the time, we decided that, computers are better than the human eye at looking at some of these things. The human eye is designed to do certain things and receive certain inputs, but it isn't exactly great at counting, especially at the level that we needed for research, where everything was, tell me if there's 200 cells in this tissue. Well, to count 200 cells actually takes longer than is really desired from a pathology perspective. It's also one of those things where things they tend to just guesstimate, and so they're looking at surface area by surface area, what cells are there, and are there 100 or there 200, and they say, well, roughly there's 200. And what we found was that 40% to 60% of the time, pathologists were off on their count. And so that revealed to us, like, hey, we need to do something that really is able to count these things fast. And it's really on the low end of a doctor's training, and they don't go to school for 14 years to count cells, right? In many respects. And so we found that they had no problem getting rid of that relationship and saying, yeah, you tell me how many you see in here, and I'll tell you if you're right. And so our system is designed really as a to allow, give answers and then let the clinician say, yes, I see that. That is correct. And I'm willing to adopt that. Or they can adjust it and say it's a little bit more or less and it missed this or that. But generally speaking, in order to look at a slide, which isn't a very big piece of material, technically speaking, could take hours just to count cells. So we've automated that whole thing. And at the same time, the machine learning has become so sophisticated and so fast, and it's heading for quantum computing, where these results can be very efficient, and therefore, clinicians can see patients much faster and produce results and get through the workload that is actually, quite extraordinary at this point, where we have only a few doctors, such as yourself, people studying hematology and other oncology, where just not enough doctors in the world to support the number of patients out there. So we're readily looking at how we can speed up that process for people in the field.
Yeah, it's really good to hear. And it sounds like you guys have had a lot of success over the past years. So kudos to you all and your team. I think one concern that some people have is sort of the idea of like self-governing AI versus non-self-governing AI. So sometimes when you keep adding more data to your system, you have to wonder, is it still validated? Is it still going to work in your population? So I know obviously we're not dealing with actual clinical side, but just from a pathology side, what's sort of the role of self-governing AI?
When we decided to use AI, we decided, we have experience with diagnostics and therapeutics in that whole process of getting through the FDA. And for the most part, we knew very well that we would be training sets of data, but then there would be, in order to get something through, you also have to have confounding data. You have to have different cohorts that, challenge the AI and say, is this actually what you're seeing? And so what we did is we hired a number of pathologists to train this. And in this case, we chose Visiopharm because they had 20 years in the industry of allowing people to train AI in pathology. And we started to train, and then we introduced a cohort of let's say 100 in a certain indication. These were pristine samples that we knew were good. We knew the diagnosis was good, maybe twice and 3 times checked by pathologists, and then they were able to train them. And what we found is that, it's iterative design, right? You let it train, and then you challenge it with new information. You let that set get bigger and bigger, and then you introduce things that are confounding, like diseases that may be close to this, to lung cancer, for example, and non-small cell, but aren't. And therefore, can it identify these cells within that situation? And so you're constantly training it, and you're making larger and larger sets. And then the AI goes in, and you start to ask the prompts and say, what do you see? What does AI see? And in this case, you can find areas and regions where we don't know what we see here. We got to re-identify that. We got to dig in deeper. Maybe we need to add some other tissues that are a little more difficult. For example, a fine needle aspirate is more of a slurry, and it has cells in it that are from the actual methodology of acquiring the sample. And so what you want to do is start to train it with those types of things. And then you get into game theory, which is really where you start to challenge it and force it to learn on its own a little bit. At that point, once its performance shows that it's statistically valid, then you have to validate it with tissue sets that are blinded, essentially. You give them to doctors, they have a read on it, then they put in their number, and then we show them what ours is, and essentially you start to have a correspondence between the numbers and say, yes, this performance is getting tighter and tighter. And we try to push that up to 98%, 100%, which is well above what is used, generally speaking, with the naked eye. And so, it's a lot of learning. Then there's the point where you have to cut it off, and you have to say, we're not going to use AI at this point. And the governance and responsibility that we have ethically and legally is that it has to perform within this certain parameters consistently every time. And although we're working in research and it isn't yet a clinical application, in order for it to get to a clinical application, it has to prove itself in the research. So we go through the discipline of validating these different data sets, challenging it, retraining it, training it again, and then we lock that down, and then we produce what's an algorithm. And so look, it's not AI at that point anymore. It actually becomes an algorithm or an application, as we call it. And then that is where a doctor, a research doctor in this case, can feel very confident that those cells that are being counted are truly those cells. And we have layers of pathologists training, but also layers of pathologists questioning these things. And we do, for the most part, we do pilots with a lot of our clients so that they can understand, is this really yielding what it is? And what we found is that, generally speaking, it's almost 100% of what it says it is. But we're very focused initially on quality control and within the tumors themselves or the cells themselves. And we set a new standard by using cellularity that is based on tumor nuclei. And so no longer estimate of that's a tumor cell, that's not a tumor cell, is the nuclei, tumor nuclei. And so we're getting a layer of detail that we've never been able to achieve before.
Thanks for sharing that, Leif. And it sounds like you all are doing a lot of work in terms of making sure the cell is really a cell, getting a human pathologist to validate the data, building, larger data sets. Maybe sort of similar question, but, last year the word of the year was hallucinations, where basically the AI is creating data that's non-existent or imperceptible to human perception. Do you have any other sort of thoughts or ideas on how we can kind of prevent that within pathology?
Yeah, I think for the most part, it is that it's in the process of bringing it into a giant data lake where you have so many different images that are from different indications, and making sure that you're training on these different sets so that you're not allowing it to hallucinate essentially in any case, and then being able to narrow it back down. I think that's in essence what has to happen. I know people think of AI and that it's thinking for itself and it has a conscience, and all of a sudden it's going to take off and we're going to have the Terminator situation. It's really, we're way far away from that right now. But it does come down to, is it good science? Are we using methodologies, and are we looking at RUP curves, and we're looking at things that tell us explicitly what we see is what is truly in those tissues? And what we found is that removes hallucinations. And in this case, we're taking a very small view of the world too, and we're not taking on everything. We're not trying to say, throw an image up there, I'll identify it for you. I'll tell you if it's liver cancer or breast cancer, and then I'll tell you if it's this type of diagnosis and what stage it is and that kind of thing. We have to take baby steps right now. We're not really ready for prime time with that type of thing. And the data lake itself is somewhat imbalanced. It's based on the procedures you can get access to. And so, getting heme patients, there's 40,000 new patients a year in hematology. It sounds like it's a lot, but it's actually not that many, thankfully. At the same time, you have to have access to those tissues and you have to train on those. And the way those access points happen throughout the world is different. And, so sometimes we have to go to locations where the care is not using neoadjuvant therapy or not having an intervention early on those tissues that could wipe out certain features, and using things that are truly, ground truth, based in ground truth that is truly the morphology of those tissues. And so we have to do a lot to stay above board with all this. And these are the things that we're constantly attentive to and, concerned about when it comes to hallucinations. What we can say right now is that we're not in the clinic, and that's for good reason. Our objective is to speed up this whole process of getting into the clinic by using digital images and data and larger data pools. But we also very much control what we're going after in order to not let it get out of hand and have a lot of misinformation being, delivered.
Yeah, no, thanks for sharing those thoughts. And, I agree, definitely far from the doomsday Terminator situation. Being a pathologist is, obviously a complex job. It takes years of training, medical school residency. The way a pathologist reads a slide can be different from beginning to end. They typically tend to scan the image and make a sense of the architecture of the slide. Then maybe they count the cells and look at the nuclear ATPI, things like that. And I was actually reading a paper last week that like the different parts of the reading that the pathologist does might be optimized for different kinds of machine learning models. So one might be CNN, one might be SVM, right? There might be different models and different parts of the step from beginning to end. So maybe we kind of combine all those different things together. So my final question, just what are your thoughts on how we can take what TriMetis is making, tCAP, some of this work pathology workflow into the clinic, and what's the kind of best way of going about that in the next decade?
Yeah, I mean, our objective is to move upstream. Like for the most part, a lot of the digital scanning is being done at the end of the process. And that is where the data is become fixed, let's say. And what our objective is to get the scanning done right away. Whether it's a clinical trial and you've consented that patient, and as soon as it gets to pathology, we capture that image in order to start to understand and follow what's in that tissue because once the data diverges from that point, it's very hard to reconstitute it all. And so we want to move upstream. We also want the utilities there to be upstream. At that point, we can look at those and we can tell the clinician, this tissue is predisposed to these biomarkers, for example. And we already have this in progress where we can say with reasonable statistical value that those biomarkers are present. Then you should test for those as knowns in that tissue. That is also the point where in research, if you know that this is going to have a ROS1 mutation or KRAS or whatever in there, not only you test for it but and get that information, but it also is the point where you can screen that patient, because what happens in research and development is that the speed to which your study and your research goes is gated by the access to this information. So if you do it at the end, you essentially have to screen lots of samples, maybe rescreen lots of samples to tell you if those markers are in it. If you start way up ahead, then you know the presence of those markers, you can confirm those, and then now that allows you to do better research within the cancer indication. So, we're trying to move upstream, and in the process, we think we can deliver data that's valuable to the clinician, to the pathologist, that's actionable, that they can then say, okay, let's test for this, or this has enough tumor in it based on nuclei that we know this genetic sequencing will work, or genomic sequencing will work. That's very important too, because you don't want to get through the process and then, appealing to the economic part of this whole thing is fail and say, well, I've spent $3,000 to test this, but it failed, right? You want to know right away, does that tissue, is it viable? Can it yield some results that are important to me as the person making calling the shots? And so, the earlier the better. And in any case, we think that the convergence of those images are going to happen. Now, the systems that are out there as far as scanners go are really gated towards this kind of old model of plugging in and scanning. We think that'll change entirely, where it'll become automated, where it comes off the H&E, it's scanned right then and there, and those results are presented to the pathologist so they can read them and they can agree with them, report on them, and then move on in the process.
Yeah, it sounds really exciting. I remember I was working in the hospital last month, and we had to call hematology on, I think, Friday at 4 p.m., which is the worst time to call any consult, and we had to get a slide confirmed just to see if someone had lymphoma or not. I think the attending had to come personally and get the actual, blood draw, run it over to a different hospital, and then get it processed so that we get a read in 24 hours. But I think all the work you're doing and all the work that's going on in AI, you definitely speed things up. And I think better, faster is better for sure. So thanks for sharing those thoughts.
Yeah, I think we're doing a lot of work. We can do better with the technology that's there, and this is technology that a lot of it intersects and exists in other sectors. And what you see from Google and Amazon and Microsoft and IBM is that they can do these things. It just hasn't been applied in this area necessarily, but remarkable things are being done in this space. And I think that it's complementary to everything doctors do, and hopefully it also takes some of the strain off the doctors of having to do things. These things run 24/7 in the background. So once the image is captured, you may go home and rest and be with your family. Those things can be teed up for you the next day, and you can have telepathology at work. You can have pathology, distributed globally so that you can get answers. And it's not 4 p.m. on a Friday Eastern time, but it's actually 1 p.m. Pacific time or what have you, that the person's reading it. And now the bottlenecks kind of fade away, and our performance becomes better. And hopefully through this whole process, also the machine learning and the information that's in these tissues allows us to actually understand more. And once we get into 3-dimensional data, which is literally 3 dimensions of tissue, but also the 4th dimension being the actual data itself, I think pathology will change and morph and become more informed, and the pathologist will have more to say about these diseases and have hopefully better therapies, better utilities, and better outcomes, right? So that's where we're headed, one foot in front of the other right now.
Thanks so much, Leif, for your time, and congratulations and good luck with all the work you and TriMetis are doing.
Thank you, and good luck with your endeavors. I look forward to talking to you again. Take care.
Appreciate it.